Yeah. These days, I'm getting a lot of questions about midges. And usually the first one is, well, what is a midge anyways? And so there's a variety of kinds. It's like, well, we have several kinds of biting midges you may be familiar with, especially if you're a trout fisherman. There's black flies in family Simuliidae. There's no-see-ums in family Ceratopagonidae. And of course, there's the mosquitoes in the family Culicidae. But I want to talk about family Chironomidae, okay? And these are then the non-biting midges. As we just heard in the quiz, the adults don't live long enough to really worry about eating anything. They don't have really much of a mouth or a digestive system. It's like, okay, time to hurry up and mate. On the other hand, their larvae, and I've got pictures of 3 different genera here, they're spending most of the life cycle is in the water, eating algae, detritus, and growing as fast as they can to then emerge as an adult. And well, not every chironomid is as elegantly patterned as this female Crocodopus that I've shown here on the left, but some very beautiful organisms. The problem is with this girl is that she's only about 3 millimeters long. And so unfortunately, most people don't actually appreciate the elegance of chironomids. And also with the larvae, they're pretty streamlined. They don't look like they've got a lot of structures on them. And so a lot of times people might confuse them with worms or something, heaven forbid. But as I said, as I want to impress upon you, they're small, but there's many midge larvae of many different species in all kinds of aquatic habitats. So unfortunately, given their small size, they tend to be overlooked. So before I go any further, I should mention what I wanted to cover tonight, and I hope that will be of interest. First, I wanted to say a little bit about the general biology of chironomids. I think we've had some good, clues about that already. And then in the middle of the talk, I want to do like an overview of a long-term research project that I have on the diversity of chironomids in urban streams in Baltimore County and city, urban and rural streams, actually, and trying to use their biodiversity to say something about the health of streams. And in this case, trying to actually diagnose what might be hurting some streams. Can we figure out what they can tell us about aquatic stressors or aquatic pollution? And then finally, I would like to say a few comments and open it up for question and discussions about the midges that have been really in the news this summer, the nuisance midges in Back River. That's not actually my formal research project, but I am a taxonomic consultant with the Baltimore County, and I'm supervising a grad student who is working on that. So we'll save that and leave that as kind of a more open discussion at the end of the talk. So try to save some time for that. Okay, good. Well, at any rate, as I was mentioning, chironomids don't get a lot of great press. Usually people just ignore them. And again, when folks are out and doing explorations of streams, it's like, well, people get all excited about the things that are big enough to see easily, and organisms that have appendages, that maybe have casings, or something that's like large and kind of impressive looking, like this stonefly that was actually like a couple of inches long. So they get all the attention, and particularly groups of organisms that are fondly referred to as the EPT. This abbreviation refers to 3 pollution-sensitive groups or orders of stream invertebrates. So the E is for the Ephemeroptera, the mayflies. The P is for the Plecoptera, or stoneflies, which are particularly need cooler water, so they would be in the same habitat you'd find trout. And then finally, the Trichoptera, the caddisflies, which come in a whole variety of different genera, and they either spin nets, as they might do in the stream up here, or they actually use silk glands in their mouth to construct cases of species-specific material. So you hear the EPT a lot when you ever hear about assessing stream health. And back in the good old days when I was working with Save Our Streams, it's like, yeah, we were happy to identify members of the EPT. So they're widely used in assessment, and those are the good organisms that you want to see. But it's a little bit sad that they do get all the press, and instead the chironomids, I think, tend to get the short end of the stick. So I want to say a bit more about them tonight. Okay, well, really basic stuff. The family Chironomidae. It's actually a very big family of small but numerically abundant aquatic flies. And the aquatic part is in the life cycle of the immatures. Eggs are laid into the water. We get a larva, which generally looks kind of worm-like. Most of them may be only a few millimeters long. If it's closer to an inch, that's pretty gigantic. They spend most of their time eating, which is why this tubular body plan is actually pretty good. It allows them to process a lot of food. And then the amazing part to me is insect metamorphosis. At a certain point, they have to transform into a completely different body plan. And for several days, instead of being a worm, they look sort of like a spitwad, but you can already see here in the pupal stage the beginnings of eyes and wings. And even more elegant is when it's time for them to leave the water. The pupa's usually been hiding out so that nothing will eat it in the water, but it has to make the dangerous move to swim up to the surface of the water. It bumps its head on the surface, and then the skin, the exoskeleton on here, splits open, and out on top of that floating skin pops out the adult fly. And if all goes well, it just ups and flies away. So later in the talk, I'll be talking a lot about pupal skins, the stuff that's left over after the pupa has successfully emerged as an adult fly, which is now ready to go find a mate and reproduce and make some more eggs. So that's the basic idea. Oh, I mentioned, yes, mating. This is, of course, as adults, that is the chief task on mind. And if you look carefully, sexual dimorphism is a big thing. It's the males, you can usually pretty easily see, have these big feathery antennae. And those are used in basically recognizing the females of their species. It's like, I understand this may be related to, say, the frequency of their wing beats. The females, not so fancy on the antennae, but you can always tell them by their body size. They're nice and husky because they have had to yolk up a couple hundred eggs that will be laid once they get fertilized. So at any rate, how do they find each other? Well, this is the part that sometimes gets notorious. The male midges do what animal behavior people would say was, they lek. In other words, the males come together and they form these big swarms, and they're basically advertising. You know, they're kind of small. They might be hard to find individually, but if they make this big swarm, usually in relation to some kind of an object at a certain height or some kind of lights, a big swarm of these males makes it a lot easier for the females to find them. And then, according to one person who analyzed love in a chironomid swarm, apparently the mated females have been attracted to the males with wing dimensions that make them more agile flyers. So you can take that where as you will, but there is some sexual selection going on here and a lot of mating advertisement. So, but once the eggs are laid in the water, and then we have a very large number of tiny little organisms. And I like to refer to chironomid larvae as the rabbits of the aquatic world. It's like they're small, as we just heard, they can reproduce rapidly, the larvae can grow rapidly, and everybody wants to eat them. So how do you manage? It's like grow as fast as you can and try to survive to adulthood. So typically, if we're looking in streams where there might be logs or rocks on the bottom, you will see the chironomid larvae have constructed little camouflage retreats. They're sort of tubular retreats here, a paperclip for size. And this rock, there's probably some competition for food here. So each larvae has its little retreat. It's sewn together, the algae and the detritus, and it's not only hiding, but it has a little pantry to munch on where hopefully no one will see it. Unfortunately, predators do find them. Here we have a dragonfly nymph, which is munching down in one of those nice bright red chironomids. Maybe the helgramide might be eating them, but as you can imagine, these larger invertebrates then will be prey for fishes, small fishes, larger fishes, and ultimately us. For those trout fishermen out there, I was pleased in the San Francisco Bay Area years ago to find a store called the Midge Fly Shop. And on further inquiry, it turns out that in trout streams, there are also midges that like that cool, clean water. And they're usually well hidden. But when they make that dangerous pupal excursion from under the rocks up to the surface, they're big and bold, and that's when the trout find them. So if you're right there at the right time and you're into tying flies, people will actually imitate chironomid pupae and use that as a fly in trout fishing. But finally, something maybe a little bit closer to our kitchens is just thinking about aquaculture. And in many countries in the world, aquaculture of fish for food is a big thing. In the Czech Republic, is kind of in Central Europe, don't have any big oceans nearby. But in that culture, actually, fish ponds have been used to raise carp ever since the Middle Ages. And so carp's a very popular food, and actually it's the classic Christmas dish. So here we're actually seeing a company, this is more like their processing ponds for carp, but they have huge ponds, they seem like lakes, that they manage, encouraging the midge larvae in them, and that is then in turn is the food for the carp that are then processed for human consumption. So as I said, rabbits of the aquatic world, small, but they grow fast and they provide food for a whole variety of organisms up the food chain. Okay, and now we've already heard something about those red midge larvae. So it turns out that certain genera of midge larvae have evolved the ability to actually create hemoglobin in their bodies. And it's like hemoglobin, like us, right? And what is hemoglobin good for? For attracting oxygen for organisms to use for their respiration. And so it turns out, particularly in larvae which specialize in living, say, in the muddy bottoms of very nutrient-enriched, algal-enriched kind of eutrophic lakes, larvae will make burrows in the sediment, actually creating these kind of tubular structures. And they're inside, and they actually wiggle and do a little dance in the tube to help water circulate in and out of the tube so that they can actually survive in this oxygen-free mud zone. So that helps bring in food. They get water with a little oxygen in it. They manage to extract the oxygen, and then in all their wiggling behavior, they actually encourage more water circulation in and out of the sediments and help recycle nutrients back up into the water column. So the chironomids that have the hemoglobin tend to be the ones that have a larger overall body size, and because they're bright red, when people see them, they're going, oh, bloodworms. And so this carries on the impression that, well, of course, all midge larvae are red, and they must all live in rather, over-enriched, polluted places. And so there tends to be this assumption that midges are a sign of pollution. And even in urban streams, a lot of chironomid midges, they're considered to be like a classic indicator of like your stream has succumbed to urban impacts. But in a way, the urban stream stuff is because, well, the sensitive EPT taxa disappeared a lot earlier. And so you're really in more stressed out urban streams, you're just seeing who can still handle it. Maybe a few Trichoptera, but some midges. So we're kind of starting out with critters that are small. People don't tend to look at them. And then when they do, they think, oh, those are a sign of bad water quality. Well, the question is, what is going on? So my tack is I'd like to, challenge the myths. And it's not just one kind of midge. I actually had some students contact me about a project the other day, and they wanted to know, what about the midge, whatever the midge issue, the midge problem. And it's like, there's not just one kind. In fact, the whole family is really diverse. Guesstimates are that there may be, easily 2,000 species in North America. And part of the fun is that it's still possible to describe previously undescribed species around here. And I have a couple that I'm looking at myself. Okay, So to dispel the myth, I just want to mention that chironomids at the species level, they vary a great deal in where they want to live, how tolerant they are. You have some that you're only going to find in beautiful trout streams. And then, yes, there are others that are quite tolerant of enriched, low oxygen, or otherwise stressed out environment. So around the world, they actually do play an important role in trying to assess the health of freshwater systems. Unfortunately, working with those larvae is not the easiest thing to do. And usually in practicality, bioassessment people, some of us tend to specialize on the midges just because it's extra work to process the larvae and some careful microscopic technique to actually see what's the genus. And then actually, so you can't really tell the species from the larvae. But at any rate, needless to say, it's a special art to identify the larvae, even to the level of genus. But there are dedicated aficionados, as I mentioned. Taxonomist John Epler's described quite a number, not to mention the Grateful Dead midge. Some of you might appreciate that. Dicrotendipes thanato-gratis. How's your Greek and Latin? Well, now that I've said midges are diverse, except that they're kind of hard to identify as larvae. So how do we get around this? And so it turns out there is a different method where instead of looking at the larvae, it's very easy to collect those pupal skins I talked about that float on the surface of the water after the flies have emerged and flown away. And so you can collect those skins just by taking a shallow pan and skimming the surface of the water, kind of like you're trying to get the fat off the surface of the turkey gravy at Thanksgiving, right? You're just kind of skimming. And that's nice because you miss a lot of the debris that would be sitting on the bottom if you were going in with a big net. So this is a way to collect a lot of these little floating midge skins quickly without a lot of debris that you have to pick through. So it's very efficient. You can tell I'm actually sampling an urban stream here. I've got gloves on. But what do you get? And I'm trying to show a picture under a stereoscope of like several different genera of midges. And it's like, yeah, you can tell them the genus just under a regular stereo microscope, which you would use probably for any benthic invertebrates. So in terms of size and structure, it's like, wow, there's a lot of things to look at here compared to the larvae. So they're easily distinguished as pupae. Also, the nice thing about panning them off the stream surface, wherever floating bits collect, is you're actually getting a more spatially integrated sample of what's in the water there. So if you were going with a net, you'd have to like jab in and try to collect in a bunch of places because the habitat is so variable. But I like to say that collecting the pupal skins, more formally known as chironomid pupal exuviae. Exuviae refers in the Greek to old clothes, cast off old skins. I'm just going to call them CPECs in case I forget, and I don't want to do a tongue twister. So at any rate, pupal exuviae. Well, as I mentioned, why mess with them? Actually, that makes it a lot easier to get genus ID and actually species ID instead of, don't even worry about the adult flies. That's even more complicated. But the nice thing is you can just look at them under stereoscope and go to genus. And if you do even a temporary mount under a compound microscope, you can usually go to the species level. So it gives you a lot of information with very little processing, no chemical clearing and fussy slide mounting and stuff like that. So it speeds things up quite a bit. And just trying to show with these 4 organisms, 4 different genera look really different. And if you're lucky, or in this case, this fly was unlucky, it didn't quite manage to get out of its skin at the surface of the water. And alas, it drowned. But for those of us taxonomists, it can be a very nice tool to confirm or to link adults with the exuviae, because after all, the adult males are still the gold standard for trying to do a scientific description of a new species, if you happen to find one. Okay, so chironomid, pupal, exuviae, why these are useful things for study. Okay, so at this point, I'd like to give a little overview of an urban stream project. And at this point, it's like I should blame Ken Belt for getting me started in urban stream midges many years ago. So thanks, Ken. So the question that I want to look at is if we use these pupal skins and try to identify them to species level, which isn't generally done. You can't do it, the larvae. But what if we go to species level? Can we get a better picture of the health of your urban streams that people usually blow off because they said all the sensitive EPTs are gone, these streams are trashed. And I'm going like, well, maybe we could actually tell better which ones are in serious trouble. And maybe other urban streams are actually starting to get better because, you know, a bunch of money was invested in doing stream restoration. It'd be kind of nice to know if a poor stream is now getting a little better, right? So maybe if we look more carefully at this diverse group of critters, and what can they tell us about the condition of streams in rural to suburban to urban areas? So specifically, I want to look at the tolerance to two different kinds of pollutants amongst these species. And maybe the chironomids might be able to help us distinguish what's the worst problem in a particular degraded stream. And the two that I can pick on, because these are things I could measure, one is a measurement called conductivity. And this is something easy to measure in the field with a little probe. But it's really, conductivity is just a way of measuring salinity or a concentration of dissolved ions in water. So like if you take table salt and you dissolve it in water, it's like, well, I don't see the granules anymore, but you've got positively charged sodium and negatively charged chloride ions floating around in there. So conductivity measurements with my little meter won't tell me what ions, but it will tell me the overall abundance of them. And as we'll find out, there's certain ions which have become very abundant indeed. So conductivity is kind of an alternate for salinity. And then also, I was looking at metals that might have corroded from human created surfaces and that have become attached to the sediment. Metals don't usually float around in solution that much, but they stick to organic matter. And so we collected sediment as well. So here's our two possible stressors. So as I mentioned then, we try to look on a study of midges across streams from very rural areas to suburban, and then to some of the most stressed out streams I could find in Baltimore City. All right. Well, this map probably looks kind of familiar to y'all. It's like, okay, thanks to Baltimore County for this nice map with the streams. The little red dots are study sites in the city and county. And once again, I want to thank Save Our Streams years ago, because thanks to their efforts at assessing stream quality, I was able to go into this and actually having their ratings of streams from very good to fair to poor to like really in tough shape. So I picked 25 streams, trying to go into it expecting that some would be good, some would be better, some would be worse, and some would be tough. So thank you to Save Our Streams for helping me kind of set up the rural to urban gradient. And then thank you to all of my intrepid undergraduate students who would go out with me to sample these streams. So again, we surveyed 25 stream sites on 12 dates throughout the season, basically from February to December. And we're trying to measure things that would matter to chironomid larvae. So stuff like, what's the texture of the bottom? Is it sandy, gravelly, rocky substrate? What's the water chemistry? Okay, nutrients, nitrogen, phosphorus, but also these sediment-bound metals. And then, of course, salt. Then we also measured things like, what's the tree canopy over the stream? Because if there's more light, you could get more algal abundance, including this really long filamentous cladophora algae, this long stringy stuff, which not only is food, but it's also a habitat for midges. So we're really trying to get a midge's eye view on habitat. And then, of course, while we were doing the habitats, we also were collecting those floating pupal exuviae. So we have a lot of those, but I'm just going to focus on what the midges that we collected for mid-June, so kind of early summer, and that gives us a lot to look at. So some fond memories of students in collecting, and actually the sofa was actually one of our more referenced streams. You never know what to expect. Okay, so we've been doing some nice approachable biology, and now have your patience and try to give you a little flavor of what we found out. It was a lot of information on habitat, and so I'm trying to summarize this, all of our streams and the things that we measured in them. I'm summarizing this in what you could consider as an environmental map here. So this little figure, this diagram, is generated to show how similar were all the streams. They're marked with their abbreviations and little blue triangles. Those are our streams, and are arranged in this map. So the streams that are closer together were more similar in terms of their habitat features. Okay, and so that simplification aside, if you start looking at this, it's pretty obvious that towards the left hand of the graph, low values on axis 1, those are the streams from rural areas. It's like maybe people know about Baseman's Run, Oregon Branch, some of those. And then on the right side, we have very urban streams. Ooh, there's Towson Run up there, Maiden's Choice, Moore's Run. Armistead Creek is kind of underneath some other pictures here, but it's like in East Baltimore is way over there. So it's like, well, we got some kind of a gradient. And in fact, although at the time, there's a lot of talk around Baltimore researchers about an urban to rural gradient, I think what this shows us is we actually have multiple urban gradients if you look inside the stream. As I'm looking at this, and these cute little red arrows here are a way of showing how the different habitat characteristics tend to change across the different streams. So like a cute visual. But if you see a really long arrow that says sand, that just means in this direction, the streams are sandier. In this direction, they're not sandy. So this D19, that's just like large gravel. So these streams are pretty gravelly. Maybe the flash floods are washing the sand out, and not much gravel here, all sand. But the really and then we have Max Clado, that long green algae that you don't want to slip on if you're a trout fisherman. Okay, that's more abundant in these streams, not in these ones. But probably the other thing, we've got this pack of chemical things, nitrogen, phosphorus, calcium, metals, and conductivity looks kind of benign. It's just sitting there in the middle. So I was looking at this and I was going like, oh, urban gradient. And I said, no way, we've got two gradients. We've kind of got a physical habitat gradient, sandy versus rockier and algae infested. So kind of what I say, a physical and a food kind of habitat gradient. And then it looks like we got quite the chemical gradient, saltier, more metals, more nutrients in the urban streams and less in the rural streams. It's like, okay, so maybe this urban gradient stuff's a little more complicated than we thought. So I hope the idea of a map to, show the relationships between the streams makes sense. Because now I'm taking the same study, but now I'm looking at the streams from the point of view of the midges that were living in there and then it emerged away. And so we see in this map, the triangles are still where the streams would be mapped, but this time it's according to 104 species of midges that were, collected over the time and 25 streams. So there was a lot of diversity in there. And the little green dots are theoretically predicted preferred habitats of the different species. And so really all you're going to get out of that, without making it too busy, is that a lot of species would really like to live in the rural streams. So they're not all tolerant. They'd really like to live in cleaner places with the forest and maybe some trout. But there are a few that can hang out even in the really, chemically impacted urban streams. So hopefully you now appreciate that not all midges are tolerant. Actually, a lot of them aren't, but we do have some tough ones that we'd like to find out more about. And it seems like the tough ones are putting up with a lot of different things here. So according to the species assemblages, so they're not all tolerant. A lot of them are not tolerant. but I'm still kind of bugged by this urban gradient thing. And frankly, the more I look at this map, I don't like it because it looks like, yeah, the rural streams are kind of, a unit, but it really looks like we've got different kinds of urban streams. Is it chemical stresses or is it something else? What's going on? So having presented this slide, I really don't like it, but I will critique it again towards the very end of my talk. And we'll get back to a different way of looking at it, which shows as you do research, you're always learning things and changing your perspective on stuff. But at any rate, so we do have these very strong patterns, but how are we going to make sense out of them? So the one thing I do want to look at, this is really busy. There's a lot of features going on here, a lot of environmental features. And in fact, somehow the connectivity doesn't even look such a big deal, probably because it got covered up by Western Run. But let's try to take a simpler look at this, something that even I can understand when I'm tired. It's like, okay, let's just look at midge diversity. How many species from the different streams are the dots? Those are the 25 streams. And I'm plotting here is what's the average conductivity, the average saltiness of the streams from like really low salt on the left to really high salt there. How many species were observed? And any given stream, we get pretty close to 50. That's not bad for just the summer. So as I mentioned before, conductivity is a measure of the amount of dissolved salts. And although the stream habitat maps were kind of like really complex, I think if we just look at the salt, the overall picture is a lot clearer. It's like, okay, lower saltiness, lower conductivity, you got more species. And it gets more salty. There's really only a few that can still handle it. So in the larger sphere of things, people may be aware, especially if you're talking with the Isaac Walton League, they're trying to alert people about the problem of too much road de-icing salt. And as I'll mention, even the state and the county are like trying to geez, we should use less of this. But even in Baltimore City, we still have these salt boxes around, and road salt in the wintertime to melt the excess snow and ice is a major source of the dissolved ion chloride. And usually the chemists measure chloride because the salt could be sodium chloride, calcium chloride, maybe potassium chloride, whatever people are using to dissolve ice. So this is going to be a theme in my talk, if you haven't guessed. Another thing that's kind of cool is looking at this data on midges, and then it turns out that Susan Cormier and her team from the US EPA did a big analysis of a survey of a ton of MBSS data from Maryland Piedmont streams, and they looked at all of the benthic organisms, the EPTs, the chiros, everybody. And they noticed if the streams had conductivity below 227 microsiemens of conductivity, that's when you could expect any kind of benthic invertebrate. It was like even the sensitive ones were okay, but when it started going over 227, then you start losing benthic invertebrate taxa. And it's kind of wild that, gee, I found out after the fact, it kind of looks pretty close to like the data we've got here. So think about that. Yeah, there's a lot of evidence of all the stressors we measured. Maybe it's just the saltiness that's a big deal. Actually, I have to go back for a minute. This is maybe a little aside. I don't know how technical you guys want to get, but you're saying, oh yeah, the salt gradient looks really nasty. But then there's this funky blip here. It's like the salt's not that high. Why are these streams so impoverished? And it turns out those are some very urban streams that and some of them are either in very old neighborhoods or in former industrial areas. And we were suspicious about metals. But if we're trying to see if midges might be responding to metals, to be honest, we need to kind of a priori figure out, well, where would we expect to see this, right? So I was able to go to a paper by some toxicologists that looked at how much of different metals does it take to start, posing a biological threat to the environment. And they were working in the laboratory, but they were spiking sediment with known levels of copper. And my little diamonds here are the copper levels of streams. Or lead, the purple squares would be a concentration of lead in my streams. And then on the x-axis, wherever those little squares and diamonds go, that's the amount of zinc. So we were looking at basically zinc, copper, and lead. And The colored lines on this graph are showing what we call the threshold effect concentrations. Like if you have, I think it's like 40 milligrams per kilogram of copper, this really low level of copper, maybe it might start to hurt organisms, but it's not entirely clear. But then you go up to a higher concentration, 150 milligrams per kilogram. That's the probable effect. Like now there's so much copper in the sediment that it probably is going to start poisoning creatures that are in contact with it. So the threshold is kind of like, well, maybe we should be worried. For lead, pretty low threshold level, which kind of makes you wonder. Think about the days of leaded gasoline. Probable effects concentration, a bit higher, kind of like copper. And then zinc's amazing. Threshold, okay, this is maybe about 150. And actually the probable effect's pretty high. So the bottom line, don't worry about the details, but any streams, any diamonds and squares that are in this kind of box, whoops, wrong diagram, the yellow box, pretty much anybody up here, there's a good chance these streams might have enough of copper, lead, or zinc that they're hurting the organisms. And so that's what I'm going to look at. I'd like to compare midges that live in the high metal streams with ones in the highest conductivity streams and see if we have different species there that might act as indicators. So I hope that kind of makes sense. So we picked out the highest metal streams. And now, quick overview of what I did. So here's some of my streams, the top contenders for highest conductivity. It's like, oh, Scott's Level, Herring Run, White Marsh, Dead Run, Towson. And we can see the conductivity values. Some of the Dead Run and Towson were really salty. And then our high metals, our group of 5, Stony Run, Moores, Maiden Choice, East Branch, Herbert's Run, and Armistead Creek were the really winner. Kind of overlapped with my conductivity. Sorry, there's a little overlap there. In fact, they kind of these guys still have kind of high conductivity. But nevertheless, I think we do have a high metals group of streams and a high conductivity group. So we're going to try and see if the midges are much different. So 33 species that were found in the high metal streams, 31 are shared with just the conductivity but no metals. But if you were going to a stream that was missing, you know, the other species and just had the tough guys, then you might wonder that some metal pollution was a problem. Okay, so tangible results. And this is pretty basic, as I say, ocular inspection. I just looked to see on my lists of midges, midge species from those exuviae, which ones were most common in the high conductivity streams, the very salty ones, and any names, these you're going to love these Latin names, anything with the double stars, those are the really common ones. We're just like, keep it simple. These are abundant, and you figure if they're that common and it's that salty, they must be pretty tolerant of that, right? And then we look at the high metal stream. You saw as a group of 5 that looked really suspiciously metal intoxicated. And it's a different group. Now, honestly, a lot of Crocodopus, you know, these guys could also be found in the high conductivity. But if they're fine by themselves and not with these guys, that's really suspicious, right? So I don't want to belabor the names too much unless you guys want to go back and talk about it. But it is interesting. Sometimes genus doesn't tell you everything. Here's a genus Nanocladius, species Spinoplenus. It can handle the salt. It can't handle metals. That's Nanocladius rectinervis, its sister species. So, something to think about. So I won't dig into this any deeper, but pretty strong evidence that the midges present in our urban streams can give us a clue. Is it getting too salty? Or maybe we have some traces of, you know, high metals from corroding siding or guardrails or brake linings or industrial things like that. So I think chironomids did give us a bit of a message. Okay, we're almost near the end here. So as I said a little bit earlier, I was showing you these maps with arrows and saying like, here, you can map, you know, and kind of visualize urban gradients, except I don't think there's like one gradient. It looks like there's multiple. And over time, I thought, why are humans always trying to come up with these ideas of like how nature's supposed to be? Of course, there should be, a gradient of land use and a gradient of stream quality. And I'm going, no, let the chironomids speak. Let the bugs tell us, oh, well, what are the streams like? And they're telling me it's not an urban gradient, it's a habitat hierarchy. So, not to go into the gory details, but if you use an analysis that's trying to let the bugs, I should say the flies, tell you which streams are more similar, it's like thinking them as clusters nested within each other. And just letting the midges tell us, they gave me these clusters. We also clustered the streams according to the midges and the midges according to the streams. And they all agreed, oh, fancy. I go back after the fact and I start mapping on some of these environmental factors and they just fall right into place. Here in this big clump, we have high conductivity streams without that much forest cover, so like suburban streams. Some of these streams have so little tree cover, they're full of that green, long, stringy green algae, and they have very particular midges in there too. Here we have in this big clump, this big cluster, are the streams with very low conductivity, low salt, a lot more forest. Some of them had a lot more sand, and those tended to have a fairly similar communities of midges. And then we have the extremist group out here. Once again, high conductivity. And this is a little group where the metals were really high, and there's really only a handful of midges that can take it. So I thought this was actually thinking of letting the organisms tell me what the habitat is like. And it's kind of like a hierarchy. So, physical features like sandy or rockiness, the kind of food, how much algae is there a lot of tree cover, and then some of our stressors, the saltiness and the metals. So I think let the organisms speak for themselves, and they tell us a pretty clear story. So at this point, I'm thinking this is my chance for a public service message for stream organisms. And I hope I've warned you, if you didn't already know about this, that, road salt is really posing a hazard to freshwater organisms and also even to humans on low sodium diets. Just noticed that the Baltimore County Council on Environmental Quality noted that the finished drinking water that we get from Baltimore City, the sodium levels are actually at, or maybe sometimes a little bit above, the EP recommendation for people on low sodium diets. It's like, ooh, where's that coming from? Ooh, salt. And as a reminder, in terms of our organisms, recommendations from Susan Cormier and group is 227 microsiemens, or else things start going downhill, and a lot of our streams are stressed out. So, of course, when I was doing my last sampling, walking back through my campus, and it's like, yup, there's salt all over the place. Sometimes in my neighborhood, there's huge piles of it in the road, inches thick. But I guess for the take home, I want to say, what are the midges telling us? For sake of time, let's just say the midges are telling us we need a new salt-sensitive attitude. I know the State Highway Administration's trying to train the counties and everybody to get their contractors to use just enough. You may see those little streaks of salt brining on the road right before we expect a snowfall. So they're trying to do better. But other studies, like in Toronto, that looked at this in detail, found out, if the municipalities are really trying to cut back on salt, it helps. But sometimes, private property owners or institutions or maybe private contractors aren't aware of the issue, and then they get a little too generous with the stuff, sometimes a lot too generous. So I guess my take home after studying urban streams, not only in the summer, but actually in the winter, is like, oh, please use just enough road salt for safety's sake so nobody slips and hurts themselves, just enough for traction, maybe even try inert things like gravel or kitty litter. But please, the midges are asking you, keep our fresh waters fresh. Normally, I would say acknowledgements and thank you to lots of students, Kevin Brittingham, Ken, Save Our Streams, Towson University, UBEL, and to my other chironomid colleagues. But I'm sure these were not the midges you wanted to hear about. So as a teaser, if I have a little time left, I'll just mention a few things about the infamous nuisance midges, which are wreaking havoc along in Back River and now other rivers up the shoreline towards the Susquehanna. As I said, I'm just the taxonomic consultant in Baltimore County, but I've got a grad student working on this. So I've known a little bit about it. The problem, I really can't do justice with how thick these things can get, but some of my colleagues are certainly filling me with ideas. Just for example, we were out, a bunch of folks, of officials were out there surveying the problem last spring. And just for example, here is one boat, and you think, this boat is filthy. No, it's not. It's a clean boat, but that's just covered with these swarming adult midges. So, and you can kind of see as Kevin's trying to catch them, they're flying around, and it's just all this brown stuff. It's just midges, live midges everywhere. And I know some of you guys know it's much worse. We know it is a nightmare for marina owners, restaurant owners, people living in houses. Periodically, these synchronized emergences come out and they're mating, but the rest of us is just like causing chaos. The weird thing is, apparently these swarms weren't really bad until about 2009. For sake of time, I'll just mention that my little piece of contribution to this is identifying, well, who are the culprits? And traditionally, before there was an attempt at control, one of the biggest culprits was this relatively large midge, Chironomys crassicaudatus, Chironomys of the big butt. Its larva is shown here. It's actually bright red, but this one's been pickled. And they like to live in the mud. And they're eating all of this decaying organic matter down in the mud. So they're pretty easy to sample, and Baltimore County is sampling there frequently. The other culprit midge, as we find out, particularly near marinas and things, is called Glyptotendipes. And it won't live in the mud. That I had my grad student and I have determined they don't live in the mud. They want to live and make their tubes on submerged surfaces. And much to our horror, pulling a boat out of there and looking at, so there's a boat on a trailer, and if you look at the hull under the water line, it's just like all midge tubes, and that's like Glyptotendipes. So it's just like, oh my gosh. So this is just in Back River, where I have more experience. And let's suffice it to say at this point, for various reasons, wastewater treatment plant, the Chesapeake Bay being a transition of urban stream to bay, whether erosion accumulated of mud, somehow this has become, since 2009, a perfect environment for at least two kinds of midges, not very many more, actually. There's really just about 6, and these are the two common ones. So that was a problem. And yes, starting about 2022, attempt was made to use an ecological pesticide, the stuff called BTI. I can talk more about that later if you want. And that's been applied in Back River on a huge scale, on a scale that no one has ever done before. And it's in an estuary where the tides go in and out and it's got flow. So the whole attempt to try to chemically control the midges in Back River is really a cutting edge thing. Nobody's ever tried to do it on this level. So the cool part was, as my grad students finding out, gee, for a couple of years, it kind of looked like the BTI was helping suppress the midges. And we were like thinking it was great. But then 2024, 2025, as many of you know well, it doesn't work anymore. And then, this year it's like, oh my gosh, it was horrible. There's like, in fact, the nuisance swarms are spreading. And to be honest, at this point, everybody's still trying to figure out what happened. So that's my tiny, tiny little overview to the nuisance midge part, and at least what I know about it. So now I probably would go back to the acknowledgments and thank yous, and hopefully I haven't taken up too, so maybe ran a little bit long, but any time for questions, or if you want to pick up on nuisance midges or anything like that, I thank you so much for your kind attention. Okay.
Thank you, Susan. You can unshare, come back. I'll put a spotlight on you. We do have questions in the chat box. Let me spotlight you here. Okay, woo, there you go. All right, thank you so much. There's so much information about so little things. I love it. Let's see.
I like microscopes.
Yeah. Jillian asked, can molecular techniques be used for identification of larvae in conjunction with environmental evaluation of water bodies?
Yes, this is actually one of the cutting edges. And in a different project, I am working on doing DNA barcoding with a subgroup of the midges, actually the Crocodopus, which showed up in the metals tolerant group. And I've also barcoded a few of the backriver midges just to confirm who we've got, because they're kind of tricky to ID. So in terms of identifying what you have, as I say, in some groups are more difficult to identify a species than others. It's useful. I think the challenge today is that people are kind of, streamlining it now. If you get an entire jar full of midges and alcohol, or all the, not just midges, like all the bugs you get out of the stream, and you just have them preserved in alcohol, can you actually take the alcohol and do some DNA processing on that and then figure out who is in there? Now, if you hire a professional molecular biologist who studies genomes, they've been working a lot on that. And I understand this method of using ways to sample the DNA right out of stream water is like really good for assessing fish and salamanders and things like that. It gets a little trickier when you get down to the level of the insects, because if they're really small and they're not common, you might not pick up the signal. And then my point of view, there's been some nice studies that actually use the barcoding to identify different kinds of organisms, including midges. I'm mostly looking at the midges, but it doesn't give you a name as to who they are. So you can't connect that with the rest of the literature unless somebody like yours truly has actually collected a known midge and then extracted its DNA, and what my colleagues would call established a DNA barcode library. So if somebody has contributed to identifying the known things, publishing a DNA signature that other people can use, then it gains a lot of value. So it's in process, and I think it works really well for vertebrates and other organisms, but that would certainly ease processing, but then you still have to have people that are good molecular biologists. So I don't know if that helped. I saw another question pop up here, something about
yeah, Susan, there's some other questions farther up in the chat too. So one from Stephen is, what is the ratio of midge larvae exuviae to other organisms such as mosquitoes?
Okay, well, it's actually I can say for mosquitoes, as long as we're not in a drought and the stream flow is pretty good, it's going to be midge exuviae and not mosquitoes. And you pick up the mosquitoes during the summer when there's a drought, really low flow, and then the mosquitoes start colonizing the stream. Now, the bigger trick is to actually relate the exuviae to the densities of, actually, you would have to be fair, where the exuviae are really looking at the adults flying out of the water. So you would have to match that with, like, for you trout fishermen, what would be the hatch of all the other things. And they do have a special seasonality to it. But if you look at just the strictly, immature bottom data that people would normally collect, with a DNet or something, it's going to vary, but the numbers of the larval midges are usually, if you're in a agricultural, suburban, or urban stream, the densities of the midge larvae are going to be a lot higher than the EPTs, dragonflies. The EPTs, dragonflies are going to have lower numbers because they're big, but also, the habitat might not be so suitable. The flip side that drives me nuts is if I'm in a healthy, beautiful trout stream and I'm looking for sensitive midges, they're a lot less common than the stoneflies, mayflies, caddisflies, who are actually, studies show, are pretty aggressive competitors, and they actually shove out the midges and eat all the food up. And so in beautiful forested, clean streams, there is still midge diversity, but they're not as abundant, probably in part because the other sensitive organisms, the big guys, are like crowding them out and taking over the resources. And it's like, well, it tells you something, doesn't it? I think you got a better view on the chat than I do. So
yeah, I'll go through it for you. Toby asks, how far does the exuvia drift down the stream, and can the mix of species reflect parts of the stream that are not relevant to the polluted ones?
That's another good question. People in Great Britain and Ireland, when they were still developing this, were actually trying to measure the drift with like little bits of sawdust, colored sawdust, And the general thing seems to be about maybe half a kilometer, but as you might imagine, it kind of depends on how wide the stream is and how many, down trees and brush there is. But when you use this technique, you're exactly right. You have to wonder what's upstream. And since I said it's integrating everything washing down from some distance upstream, the exuviae technique is really best if, like in my study, you're trying to compare independent, very different streams that aren't interconnected. So yeah, you have to think about the drift. Actually, a long time ago, Ken and I actually did a study where we were looking at the benthic invertebrates above and below parking lots. And so the exuviae technique would not have worked there at all because everything would have washed in and been a mess. And then you have to use the benthic stuff. But I'm arguing if you're trying to do a big survey of a whole bunch of not connected streams, the floating stuff can work pretty well. But again, if there's a big quarry or some kind of, stormwater outfall upstream, you're going to have to take that into consideration because that is going to influence what you're finding. Hopefully that was helpful.
And then how far do the adults tend to disperse? Do they keep to the same stream or do they fan out over a large area? And how widespread are most species? Are they spread over narrow region or continent, global, or how?
What a great question. It's kind of, we still argue about that, or I should say speculate about it with the backriver midges. And that's why trying to identify different species is helpful because at least sometimes we can eliminate certain places as sources. But it seems like locally, they're not super strong flyers. If it's a windy day, they may not want to fly at all, or they might get blown around. But I can't imagine that most of them probably get blown around much more than a wild guess, maybe, a kilometer or two. But occasionally, windy day, some of them get further. And the flip side, you mentioned about some species on really large scales. One of my other pet projects looking at DNA barcoding of the genus Crocodopus in the Northern Hemisphere. There are species that are common around here, like Crocodopus bicinctus. It's pretty much all over the Northern Hemisphere. And my DNA barcoding projects are helping me look at the genetic variation, just with one gene, really, even across North America, comparing like Crocodopus sylvestris. You saw that cute, colorful little female fly at the beginning. Slightly different genetic signatures, but basically the same thing. Western Europe, North America. It gets a bit different by the time you get to Japan and China. It's looking kind of different. But on a larger time scale, the common, how should I say, adaptable, somewhat more tolerant ones can have a very large range, like, transcontinental. And then some of them are very specialized. And like there's a study from the rainforest in Australia. They found some species that they're just like in this one stream. They don't get very far at all. So once again, it depends on what species you're talking about and how specialized and sensitive versus how robust and tolerant they might be and how far they can get.
Have you worked with any chironomids present in lentic systems?
A very long time ago. Actually, in a wetland in Illinois at Argonne National Lab, I was looking at competition between different groups of aquatic invertebrates and midges living on these, basically wetland plants were abundant and not very good competitors with snails. Actually, the snails beat them up. Everything bigger always beats out the midges. Also did a foraging study in lakes. But those were really focused experimental manipulations, but they weren't really survey kind of data. So ever since the California, I've really been looking much more at, big scale, at surveys in nature and that. So not as often in experiments. So that we've done some experiments, but on very small scale.
All right. Ken says, some of us are curious about Dead Run because that's where we go and play in the stream together. And in your cluster analysis, it's sitting all by itself. Do you know why?
That is fascinating. And actually, in trying to barcode more of my local Baltimore midges, I'd love to look at dead run a bit more. Uh, really high connectivity, and I guess probably there's a lot of major highways, roads nearby in the watershed, and so it just looks like it gets a lot of salt runoff. And actually, this is right by the USGS gauging station. Ken probably knows the place I'm talking about. But you could also tell that when there were rainstorms, the severe, short, sharp flooding events that are common in urban streams were a big deal too, because the banks were really cut down, and if you walked in the stream channel, the rocks and the sand would kind of roll around under your feet. It's kind of creepy. But I think that's because, the storms were, even small rainstorms, with all the water rushes off, it stirs up the bed. And so the rocks never really settled down that much. So I'm thinking they were probably species A that can, tolerant of salt, but also things that have many generations and that can repopulate rapidly for the disturbance, or maybe they're really good at digging into that soft sediment to stay away from the storms. But it's interesting because it's one of my more extreme streams, but it's so different from the metals groups. It's really fascinating. So maybe physical factors, the saltiness, and I hate to say it, but we talked to some folks that we met out sampling there, and they said that they would go hunting for crayfish sometimes, but sometimes they got allergic reactions from the water. It's like, I don't know what else is in there, but sometimes we would meet people from, in the neighborhood, and they would tell us stories about what they knew about these streams. And it's like, that's why we're wearing disposable, nitrile gloves. So yeah, that's a great question, but believe me, the work still continues. I'd ultimately like to refine my indicators in a more formal way. And also, I definitely want to keep going through samples for other time points, some early spring stuff, and maybe get a bigger picture. But the combination of salt and flood disturbance seems to be big deals going on there, but there might be other mysterious things. Maybe we should check out MDE and see if they have any more information. So I hope that was helpful. Interesting spot.
Yeah, well, it is an interesting spot, and we'll let you look at all the stuff that we've found there.
Yeah, it would be nice to cross associate who's living there.
Yeah. Kathy says, are you aware of the huge unregulated fishing of mud shad and carp in Back River? They're bottom feeders, so are they no longer eating the midges as in prior years?
That has come up ever since we did a public information meeting back in 2009, and it still comes up. It's like, yeah, apparently, I guess there are some fishermen that are, I don't know, quasi allowed to do it, and I guess maybe more that aren't. And the rumor that I heard, and see if this is correct, but it's like the people that do fish them somewhat legally can't sell them in Maryland because there's so much PCB in there from people dumping it in the past. That you can't sell them in Maryland, but somebody was saying like, oh, they ship them to New York City and sell them there. I don't know.
So that ugly rumor, yeah, but they're actually completely unregulated. There are no fishing regulations on mud shad. So I'm talking to the senator, state senator, about and looking at fishing regulations to see if we can't get some at least some regulations in Back River.
That would be my first recommendation, honestly. I mean, we're trying to think out, like what happened that we can't control them now. And I'm thinking back, it's made me think about those fish ponds in the Czech Republic. And they're getting food out of these things and actually helping the midges grow so they can harvest more fish. And I'm thinking like, well, carp aren't really fussy about habitat. They should be there. And if they were eating them, it should probably drop it down. I think my take is they improve the efficiency of the wastewater treatment plant. So now it's, and now there's more oxygen, and now the midges are eating all that old accumulated organic matter, and nobody's eating them. There's hardly anything else there. But if you had, mud shad that were eating them, that's got to help. And I mean, they don't reproduce that fast. That would seem like the first thing you'd want to do is encourage whatever kind of bottom feeders you could get to go around in there, and then not have people eat them because I am also told by my colleagues that, some individuals used to stockpile leaking containers of PCBs near certain parts of the shore. And so you don't really want to eat those fish anyways. So I think this should be a great place for the fish to eat them. And then hopefully that would help. I think it's like a public health emergency.
Karen Wynn and I are both the one. She's the one that's doing the later chats here. She's with Karen with Weaver Marina. Right, there you go. And we both sit on the Baltimore County Commission for Environmental Quality. So we're all about road salt for sure.
Absolutely. Thank you.
And then I was, my family helped start Maryland Save Our Streams back in the day. So we probably ran into each other many years ago.
Great.
Well, thank you for your all your work on this.
Welcome.
Okay, Sarah asks that you wants to know, you measured sediment-bound metals because the larvae, is it because the larvae eat the sediment or because it has higher and more easily detectable concentrations?
Both reasons. And again, looking through like Baltimore City, water quality reports, it's like you don't pick up any metals in the water column. It's like they just chemically adhere to organic matter and stuff. And so the concentrations are very measurable in the sediments. Even if you go out to like the streams draining Soldier's Delight, it's like, well, there's not that much human induced metals out there, but you can definitely pick up the kind of inert natural levels of chromium, for example. So the sediments are, chemically binding a lot of stuff. And then, yes, that's where a lot of the midges are living right in there and they're eating it. So it seems like there's as much exposure as you could want to have. And so basically, I would had a method of bottomless buckets to collect sediment, subsample sediment from a known area of the stream bed. We'd bag a subsample of that, measuring all our volumes and areas, take it back to the lab, basically dry it out, and then do a chemical analysis per weight of dried sediment and the metals. So fortunately, chemists at Towson already had ways worked out for how to do this. So my students and I were able to carry that out. And it is, I guess I was afraid that it would be much more prevalent in terms of, like the really highly concentrated metals. It was a small number of streams. But if you looked back at the graph, there was actually that threshold, concentration. And it's like, well, the threshold was invented in the lab. What does it mean in the field? But I think the more natural vegetation buffers around our streams, the better. That's all I have to say. But I guess I was relieved that the metals didn't seem to be an even bigger immediate threat, but it really looked like the salt was coming through.
All right, Jillian speculates, if you could identify the larvae from substrate sampling by the molecular means, then maybe you could evaluate how one stream changes over distance, and that's not very possible with the drifting exuvia.
Yeah, exactly. I would just use the larvae. And again, I could mention that paper that Ken Belt and I did that got published. What was that, Ken? That was like 2007 or something in Hydrobiologia. That's pretty much what we did for 2 different streams. And it was relatively short. We had a couple sites above a major parking lot and a couple sites below and 2 different streams, like in Green Spring and then up in Cockeysville. And yeah, the larvae were entirely sufficient to show that there were big changes right below the parking lots where you got the flush of water, you got runoff of grease, presumably metals from brake linings and galvanized rails and stuff like that. You could see it. In that case, when we submitted the manuscript, it was a little fun because you could tell some of the reviewers actually worked on chironomids, and they were going like, well, how do you know that you actually got, the species right, the generant species? It's like, if you carefully prepare their little heads on slides, you can take them to, groups of species within genus. And they were all after me. It's like, how do you know you actually got this right? You need to have the pupae or the adults. And it's like, oh my God. So I actually went and collected them and reared some and went through and learned how to identify pupal skins from midges that I had reared in the lab, darn it. So they were like my little pets, well, until they got pickled. So it's basically you've made a good point. If you're going to work with the exuviae, somebody somewhere needs to have assured that they know what species the particular pupal skins come from. And fortunately, a lot of the common stuff has been done, but there's still work to be done. Actually, one of my Irish colleagues went through a collection from Bill Kaufman, who was a retired professor from Pittsburgh, University of Pittsburgh, and he has had this huge collection of pupal exuviae slides. So Peter Langton's just done kind of the species level guide to midge skins from North America based on this huge collection. And a lot of them are connected with an adult, like you said, or either a larva adult or something, so that you can tell what it is. And I barcode some of these things to confirm it. Other ones haven't been associated, and we still don't know who they go to, or if it's a new species or one that we just don't know much about. So you have identified an important point. Depending on your question about streams, you need to pick which life stages are the ones to look at. And in some of your questions, you might actually, as someone else was asking, you want to know what's the relationship between the other orders of invertebrates or the predators, other competition. You might want to know about those EPTs and the midges together. So there you're just going to look at all the immatures and not worry about the pupal skins. But it depends on your question. As I say, different, independent streams scattered around. The pupal skins actually save a lot of work, but as you have pointed out, you need that prior knowledge of who they go with to make it fly, so to speak.
Well, and Sarah was letting you know where our site is 2.5 miles downstream of the midge site, which is out of the full suburban area. And we have lots of forest cover. And we get lots of the EPTs in our collection, and not a lot of the chironomid things, if I would say. So they could be that predator-prey relationship going on as well. So
absolutely, predator-prey relationship. You've got a lot of fish eating them. You've got all those dragonflies, hellgrammites. Actually, even some midges eat other midges. So lots of predators, and then even those mayflies and kind of the more algae scraping, stoneflies or whatever, snails. They're gobbling up the food faster than the little tiny midges can. So yeah, you're absolutely right. In a clean stream without a lot of pollution, it's all the bigger guys, the nice sensitive taxa. They're just going to crowd out most of the midges unless you've got some, hypersensitive midges, but they're never going to get really abundant because they just can't throw their weight around like the other ones do.
All right. Do we have any other questions? And I echo a lot of people are they've learned so much. Thanks for a great presentation. Any other questions for Dr. Gresens this evening? It looks like we might have to have you back because we have to learn some more from you.
Well, I'm pleased that people learn stuff and they found it engaging. And I always encourage, it sounds like you guys are already going out to explore streams as it is. So let's all do more of it. Thank you so much.
Do that. And I love, and what I'm taking back from this talk, I'm taking back a lot of things, but I love your quote on, we're going to let the midges tell us what's going on. We'll listen to them. Yeah, so that was great.
Organisms. Yeah, way to go.
Yeah. Well, thank you all for joining us. Remember that next week it is the evolution of venom. So I hope that you'll join us back here on Thursday evening for Must Learn Thursday. Until then, stay well, stay curious, stay outside, go look, keep looking under those rocks and in the sediment, and we'll see what we can find together. Take care, everybody.
Right. Okay. Good night.
Good night. Great seeing you, Susan.
Yay. Good to see you.
And Mike.
Woo-hoo. Be well.